Hello everyone, I hope you are doing well. I am Bhagyashree and we are learning immunoglobulin structure and function. So we are going to continue with the lesson. Those who have any doubts, feel free to ask in the comment section below. Also, if you have any suggestions for the preparation of any specific topic or subject, you can write me in the comment section below. And if you like the video, don't forget to click on thumbs up button. Also subscribe for more video updates. Now, today we are going to learn about the different class of immunoglobulin. So in mammals basically there are three classes of antibodies. They are IgG, A, D, M, E. So these are the five classes. And there is a subclass for IgA and IgG. So the for human IgG subclass, it has got each class with its characteristics except with the variable region. And 90% of homology in amino acid is there for every class, but for IgA and G, it is only 60% homology. Now, first coming to IgG. So IgG class, IgG serum, 80% of the total serum Ig, and it has got four human subclasses. Differences in Y chain and numbered with decreasing average serum concentration like the IgG 1, 2, 3 like that. So IgG 3 it has got half-life of 7 days and IgG it has got the half-life for 23 days, which is the longest half-life of all immunoglobulin isotype, and it passes through placenta. And what is its function? To allow the mother to transfer immunity to fetus. So this is it about the IgG. Now coming to IgM. So first class of antibody by developing B cell is IgM. So remember that it is the first class of antibody which is developed by B cell. Then it has got 5 to 20 10% of the total serum Ig. So it constitutes 5 to 10% of the total serum of immunoglobulin. And where it is found? It is found on the surface of mature B cells together with IgD, where it serves as an antigen specific B cell receptor. Now, the molecular weight for IgM is 1,80,000 as a membrane bound antibody on B cells, and it is secreted by plasma cells as a pentameric molecule which is composed of 5 units. Now, each of which consists of 2 light and 2 heavy chain, and it is joined by an additional disulfide bonds between their Fc portions and by a polypeptide chain which is called as the J chain. Now, J chain it is synthesized by B cell or plasma cells, and half-life of IgM is 5 days. Now, the first class of immunoglobulin it is produced by a primary response to an antigen, and the first immunoglobulin it is synthesized by neonate. So it is more efficient than IgG in activating complement, and the complement activation it requires 2 Fc region in close and pentamer structure of IgM. It completes the requirement. Now coming to IgA class. Now IgA class it constitutes total of the 10 to 15% of the total immunoglobulin in serum, and external secretion like breast milk, saliva, tears, mucus of bronchial, genitourinary and digestive tract it has got. Uh, then its function is external secretion, and it contains two subclass IgA1 which is 93% and IgA2 which is 7%. Now IgA it has got a half-life of 5.5 days and IgA is monomeric. So it is also called as a secretory IgA, which is it consists of a dimer which is IgA of external secretions. And then there are polypeptide chain which is called as secretory component. Then the secretory IgA, what happens? It is transported through epithelial cells into the intestinal lumen by an IgA specific Fc receptor, which is also called as poly Ig receptor. And the integral membrane, it is integral membrane glycoprotein. So the colostrum of milk in nursing mother, it protects against pathogen for few weeks in newborn. So because of the this secretory IgA, which is transported through the epithelial cells into intestinal lumen by IgA specific Fc receptor called poly Ig receptor, and which is a integral membrane glycoprotein. IgA, it has got function for colostrum of milk in nursing mother protection against pathogen for few weeks in newborn. Now coming to IgE, it mediates the immediate hypersensitivity reactions which are responsible for symptoms for hay fever, asthma, hives, and anaphylactic shocks. Now what happens? It binds to the receptor of Fc receptor on membrane of blood basophils and tissue mast cells. And then there is a cross linkage of receptor bound Ig molecules by antigen induced degranulation of basophils and mast cells. Then there are a variety of pharmacogenically active mediator which give rise to allergic manifestations. So IgE is degranulation necessary for antiparasitic defense. Then coming to IgD, it constitutes 0.02% of the total serum, and together with IgM, major membrane bound Ig, it is expressed by mature B cells, and it has got a function in activation of B cells by antigen, and it as such it has no biological effect identified yet for IgD. So this is it for today. Stay tuned for more lessons to come. Thank you for watching.